So
I'm just going to say a few words, Gary, and then I give you the floor. A very good afternoon, everyone. You see that our colleague Garry Scarlett, Dr. Garry Scarlett, is very eager to start his presentation. So again, very good afternoon, a very warm welcome to our webinar. Today we are delighted to welcome our colleague from the University of Portsmouth, Dr. Garry Scarlett, who's going to address the question of ancient DNA. You all know probably that necromancy, and Gary is going to say much more than I do, is an art of divination. It's a very old practice based on the study of dead bodies. This is something you could find in literature from the Odysseus to Middle Age literature and obviously today as well. So we'll see whether Gary with his DNA analysis is a sort of modern shaman of today. Just to introduce Gary studied molecular biology at the University of Portsmouth. He's a specialist in his field for a number of years. He's also the associate head in the School of Biology at the University of Portsmouth, a quite challenging position at the moment, as you can understand. And his interest is in molecular biology, as you can imagine, but also biochemistry, nucleic acids, and the study of ancient DNA and bones. I must say, I visited Gary's lab and I could see a number of bones there, which were nicely stored in boxes. It's quite impressive. So Gary, without further ado, I'd like to give you the floor. A few rules before we start. Please mute your mics. If you don't want to be seen, switch off your cameras, and I will manage the questions through the chat box. Gary, the floor is yours.
thank you. So ancient DNA is modern necromancy. So science is magic, really. Every day you encounter magic in your life. We just call it science. And the type of science that deals with necromancy, necromancy is magical connections with the dead, is ancient DNA. We can connect with the dead by studying their DNA. So we're going to talk about that today. Cool. So what is ancient DNA? Well, DNA can survive for thousands of years after you die. And that can be a few hundred years. In the case we're going to talk in a minute about the Mary Rose, it could be tens of thousands of years when you discuss things such as the Neanderthals. And there have been extractions of DNA from some samples which are hundreds of thousands of years old. So when the individual, the organism, is long, long dead, that DNA, which in many ways is that individual, lingers on. There's like an echo of them down time. And we can recover that DNA that come from a long dead individual from a number of different sources. It can be from their bones, it could be from their artifacts, if those artifacts were made with organic material, but particularly from bones, particularly from certain bones. And the technologies have been around really since the 80s to do this. So that horse-like, weird-looking animal is a quagga, and it's a kind of cross between a zebra and a horse. It's an extinct species now. It died out within the last 100 years. So the first time any ancient DNA work was done was some remains of the quagga back in the 80s. And technologies moved on very rapidly. And then that guy you see there really changed the field. I imagine at some point he will win a Nobel Prize. So that's Svanzi Pablo, and he did some amazing work. He revolutionized the field of ancient DNA. We'll talk a little bit more about what he did later. But why would we bother looking at ancient DNA? Why bother going to all the hassle of finding some old bones in a pit, going through the complicated and, to be honest, very difficult process of extracting, analysing and interpreting that data? Well, a whole host of scientific fields have developed which can be helped by ancient DNA. And probably the most obvious one is archaeology. If we can get the ancient DNA from an individual, from their bones, we can determine physical characteristics about them. Their genotype, their DNA, determines their phenotype, what they look like, and to a certain extent, how they behave. So that's their phenotype. By looking at the genotype, which we can observe from the ancient DNA, we know things about them, what they look like, and weird stuff. Did they like Marmite or not, for instance? We can look at how species or populations move over time. You can study how the human population has come out of Africa, spread through Europe and Asia, commingled with already other existing hominid species. So that's population and evolutionary biology, and that's true for people, human populations, and animal populations. Related to that is the field of anthropology. Who are we? Why are we here? You know, a really deep question. And Stanley Pablo's work, that guy I showed you earlier, really shed some light there. He really revolutionized how humans see themselves, where we come from. So the fields of anthropology and paleogenetics, as we now call it. And there's another one that's starting to emerge now, and that is health implications. If you can study a population over time, if you can go back and extract DNA from our ancestors, and you can look at what kind of genes they had and how those genes are changing over time, that maybe can give us some clues about our health implications for the future. So there's a range of different disciplines which are looking at ancient DNA, which make ancient DNA a good thing to be doing. So we're going to be using a case study here, some work that we did quite a few, well, 5 or 6 years ago now with the Mary Rose Trust. So as Layla said, I'm a molecular biologist and a biochemist, and I didn't start off with an interest in ancient DNA. I have an interest in what's called nucleic acid structure and gene regulation. But I also have an interest in dogs. I've got 3 small dogs myself. I have quite a strong interest on canine welfare. And I had a friend at the Mary Rose, and he knew 2 things about me. He knew that I worked with DNA, and he knew that I liked dogs. And he come to me one day and he said, there's a dog sitting in the museum that there's a big argument about. We've got quite a complete skeleton, and here it is. This is what we call Hatch the dog. The dog has caused some controversy. What type of dog is this? Now, there's, if you know anything about dog breeding, there's an intense amount of rivalry between dog breeders and their different breeds. And one dog breed association said, this is a Manchester Terrier. There's no doubt about it. This is a Manchester Terrier. And no, said another breed association, that's absolute rubbish. This is a Whippet. This is a Whippet. And there were a few people who said, no, it's some kind of other type of terrier, maybe a Mastiff crossed with a terrier. So all these dog experts arguing about what type of dog this was based upon its skeleton. So he said to me, you know about DNA, you like dogs, can you tell me what type of dog breed this was? And in my ignorance, I said, sure, I can give it a go. I didn't realise what I was letting myself in for. It took 2 or 3 years to finally solve that problem. So this dog was the ship's dog on the Mary Rose, the ship that sunk in Solent 500 or 600 years ago. So at the time, dogs were considered the best rat catchers, and cats were considered unlucky. So ships often had a dog, which were usually associated with one of the officers, whose job it was to catch rats and small vermin. This dog was found when the Mary Rose was recovered, and it was found in the hatchway of the carpenter's cabin, and it may well have belonged to the carpenter. He may have been the main carer of the dog. The dog had got caught in the door had slid shut on the dog as the ship sank, and the dog had drowned. Because it was found in a hatchway, the Mary Rose are called the dog Hatch. So going back to what are we aiming to do with ancient DNA in this particular case study? Well, the Mary Rose wanted to know 2 things about Hatch. They wanted some phenotypic information. Now, they've got a fairly complete skeleton, but there's some things you can't tell from a skeleton. What coat colour was Hatch, for instance? The second thing they wanted to know was to resolve this argument between the dog breeders. What type of dog was Hatch? Was he a Manchester Terrier? Very cool looking dog, a Manchester Terrier, actually. Was he a Whippet? I used to have a Whippet myself. They're nice dogs. Or was he a Terrier Mastiff cross? So let's see if we can use ancient DNA to solve those questions for the Mary Rose. So this is a zoom in on Hatchie's head, and you can see he's still got some teeth. In fact, he's got one less tooth now because we pulled one of those teeth out to use as a source of DNA. DNA remains in bones. It's actually very stabilized by the matrix of the bone. And you can extract DNA from any of the bones in the body, often used are what we call the long bones, the bones in arms and legs. A better source of DNA, though, is the DNA extracted from teeth. Now, you can get up to 10 to 100 times more DNA from a tooth than you can from a long bone. So we went through a tooth here. In fact, you can get even more DNA from a very special bone called the petrous bone, which is in the skull. It's part of the inner ear. It's like a very, very dense bone, But to get it would require some damage to the skull. So we didn't want to do that. Okay, so we decided to go through a tooth. So we pulled a tooth out from poor old Hatch. He didn't have many left. Now, this is nothing to do with molecular biology. This is nothing to do with ancient DNA. I just think it's really cool. So we said to the Mary Rose, we're going to have to pull a tooth out from Hatch. And the Mary Rose went, oh, he's on display. We don't like that. So we said, I'll go to the university dentist and we'll have a false tooth made for Hatch, right? So I knew nothing about dentistry, so I kind of like this. So this is the university dentist. We share a dental school with King's College London. So this was done actually in the King's College London lab. And here they've taken a mould of the tooth we pulled out, and you can see there in white, and then they hand painted it to an exact replica of the original tooth, including all the decay and the build-up of calculus on that tooth. So it's amazing. It was an amazing thing. And they slid that back in there. So you don't know they're taking the tooth out. But the dentist did point something out. Hatch, the dog, had dental decay. The only reason the dog could have got dental decay if it was eating something quite sweet. If it had been living on a diet of rats only, whatever it could scavenged, it wouldn't have dental decay. Dental decay of this type is kind of unique to processed foods. Somebody was looking after Hatch. He was more than just a ratter. He was clearly a pet. So now we're going to move back to DNA analysis, ancient DNA, molecular biology. So if you look at the picture that you can see on your right of the double helix, if there's a secret to life, to the mystery of life, it's probably DNA, and it's that image you can see there, right? Now that image is known worldwide, the double helical structure. If there is a secret of life, it's a very badly kept secret because you can see it on this screen. To the left of it, we've zoomed in a little bit on that double helical structure, and you can see that there's what we call bases bound together, T bound to A, C bound to G. And the order of bases, as you march down that double helical structure, encodes information. And that information encodes our phenotypes, us, what we look like, to a certain extent how we behave, and to a certain extent our flavours and tastes and illnesses. What's amazing about this is that every living thing on the planet is written in the same code. There's no variation, there's no French and English and German. It's all the same language. Every living thing, from a bacteria to us, is written in the same code. There's a really sort of profound aspect about that every living thing on the planet is probably related if you go back far enough, which is a very strange thought. What's probably less known, though, is that in all of our cells, we have two types of DNA. We have what we call genomic DNA, and that's the picture you can see at the top. That's a condensed chromosome, and that has the bulk of the genetic information, by far the bulk of the genetic information. And that one encodes most of what we are. But there's another type of DNA in all our cells called the mitochondrial DNA. That's represented by the lower picture. It's much, much smaller than the genomic DNA. So the genomic DNA, there's like 3 billion bases. The mitochondrial DNA, there's only 16,000 bases. So much, much smaller. But there's something weird about it. What's weird about it is where you only get one copy of your genomic DNA, you can get dozens, sometimes hundreds of copies of your mitochondrial DNA per cell. And that has a profound consequence for ancient DNA analysis. It means the mitochondrial DNA survives much, much better than genomic DNA. So many of the early experiments, and included some of the stuff we've done here, focused on mitochondrial DNA simply because it's much, much easier to work with. Now, the other odd thing about mitochondrial DNA is that you only get it from your mother. It's only maternally inherited. So for your genomic DNA, you obviously get 50% from your father and 50% from your mother, which is why I look a bit like my mum and a bit like my dad. Your mitochondrial DNA only ever comes from your mother. And some of you, if you've read about DNA and ancient DNA analysis, may have heard of a concept called mitochondrial Eve. We won't go into that today, but that's a consequence that the mitochondria is only maternally derived. However, the problem with mitochondrial DNA is that it has far less phenotypic information. It doesn't really contribute much to what we look like or how we behave, not like the genomic DNA. So we're going to actually look at both mitochondrial DNA and genomic DNA, and we'll come to that later. But first, I just want to cover some of the problems that we have when we deal with ancient DNA. And this was new to me when I started 7 or 8 years ago with the Mary Rose. I thought it was going to be the same as working with modern DNA. There are a whole host of extra problems that we hadn't thought about. I had to do a lot of reading in the field and ask a lot of questions. DNA is a robust molecule and it can survive for tens of thousands of years, but it will degrade. So those 3 billion bases, instead of being in a long chain, get broken up into tiny fragments, only hundreds of bases long. Because you can only recover a small amount of ancient DNA from your bones, contamination is a huge problem. So some of the early ancient DNA work done in the late 80s and early 90s were embarrassingly retracted later because they were in fact contamination from the worker, from the guy who had done the experiment, or sometimes from the archaeologist who'd recovered the bone. So when we get the bones, we have to clean them intensely. We have to bleach them, UV irradiate them, and when we work with them, we have to be dressed up like this. So this is the ancient DNA lab where we extract the ancient DNA. So you have to wear a full body suit and face mask. Well, I don't ever go in there. The technical support do. So now these are just gratuitous pictures of machines in my lab because I like them, right? So the key machines we're talking about here, though, are the polymerase chain reaction machine, which is called a thermocycler, and the DNA sequencer. So once we've extracted the DNA, we can only extract tiny amounts of DNA. And that was always a problem back in the 80s for ancient DNA work until somebody invented PCR. PCR allows us to amplify very small amounts of DNA at very select locations to lots of DNA. And somebody won a Nobel Prize for that, a guy called Kary Mullis. He went a bit mad after he won his Nobel Prize, actually. Then we can take that amplified DNA from by PCR, and then we can determine the order of the bases. We can read the code. We can read that DNA code. And we use a technique called Sanger dideoxy sequencing. Sanger was probably the single greatest biochemist of the 20th, probably the 21st century. And he won 2 Nobel Prizes. One of them was for the Sanger dideoxy sequencing method. He died a few years ago, very sadly. So once you amplify the DNA and then you sequence the DNA, you'll get something like this. This is the output from one of the machines. It's called a chromatogram. And you get various peaks. But as you can see along the top, you can see the order of the bases, CGAT, GAA, GAA. And then if you look at the first black arrow, you can see something very strange. You see a red peak, and just below it, at the same position, a black peak. That means there's two bases there. Our sample from Hatch was heterozygote for what we call a SNP, short nucleotide polymorphisms. So just a quick background here. We've got 3 billion bases, right? And the far enormous bulk of those bases are identical between every human on the planet. But there are a few million places where we vary in those 3 billion. And it's those tiny, tiny single base differences that give you the massive variety that you see within a species, the massive variety that we see when we look around us, when we walk down the road. Same with dogs. So these places where our DNA can vary and affect the phenotype, we call SNPs, short nucleotide polymorphisms. And what we're doing here is we're looking for SNPs to determine the phenotype of Hatch. In fact, in particular, the coat colour. So we did this, and we looked at various bits of the DNA by PCR and Sanger sequencing to help find out Hatch's phenotype. Locate the SNP, read the DNA, determine which SNP it is, you know what he looked like. Well, he had brown fur. He had a light reddish brown fur. We can tell that from the DNA. Unexpectedly, he was a carrier, but not a sufferer, for a genetic disorder you see in some dog breeds today, hyperchorusia, which affects their kidneys, basically. He was a carrier for it, but he wasn't a sufferer for it. And this is quite interesting. It kind of suggests that that genetic disorder is very, very old. But we haven't answered one of the questions that the Mary Rose had asked us. We said, okay, he's got brown fur. We can even tell you something about his genetic health. But what breed was he? So, okay, we'll have a go at that. So we looked at the mitochondrial DNA, and we drew this as a kind of phylogenetic map, what's called a phylogenetic map. And on the little circles are different dogs and, you know, how they relate to each other. So if you sample several hundred dogs, we sampled several hundred dogs and we added them to a database of several hundred other dogs. You hope that all the Great Danes will be in group A, for instance, and all the Shih Tzus, I've got a little Shih Tzu down here beside me somewhere, will be in group B. It doesn't happen like that. It happens like that with humans. It doesn't happen like that with dogs. And the reason it doesn't happen like that with dogs is because of the weird breeding patterns humans have enforced on dogs over the last 10,000 years, particularly the last 200 years. You could have two breeders. They could be only 20 miles apart and they could breed the same breed of dog. And I actually know of a case like this. But because they hate each other so much, they won't let their dogs interbreed. So there are dogs which are in America, which are more similar to the dogs in Oxford than the dog in the neighbouring county. Because humans have controlled the breeding and disturbed the equilibrium of the breeding, you can't use mitochondrial tracing, because that's the standard approach we would use for humans. We can't use mitochondrial. We tried to put this into paper, we sent it off, and they come back and said, we like your phenotype work, the mitochondrial work is rubbish, do it again in another way. So we did. We went and used an approach called short tandem repeats. Now I've got to give you a little bit more science again, a little bit more science about DNA. You've got 3 billion bases. That's a lot of DNA. But only 3% less, probably nearer 2% of that DNA is actually what we call coding, actually contributes to your phenotype. Huge chunks of your DNA are what used to be called junk DNA. They don't seem to do anything. One of the types of junk DNA is called SDRs, short tandem repeats. And what they are is the same sequence of DNA repeated over and over and over again, thousands of times sometimes. For instance, here in this example, the sequence CAG in red. In the first individual, it's repeated 5 times. So it goes CAG, 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 CAG. But in another individual, it's only repeated 4 times. And in a third individual, it's repeated only twice. Now, SDRs are the basis of genetic fingerprinting, DNA fingerprinting that the police service use to catch criminals. And it works because there's a variety of numbers of repeats of SDRs between individuals. So we can take that technology, that approach that's used by the police service to detect criminals, and try and use it against Hatch. We can look at Hatch's SDRs, these are on the genomic DNA, not on the mitochondrial DNA, and then compare that against a database of dogs, the SDR database of different breeds. And in fact, this method is the method that is used in dog crime detection. So that's why we're pretty sure it's solid. So we did that. And Hatch is a Jack Russell. Now we have to be very careful here. 200 years ago, beyond 200 years ago, dog breeds as we know them didn't really exist. That whole dog breeding thing is very recent. What there were archetypal type dogs. There were big guard dogs. There were small terrier hunters for small game like ratters. There were herders. Hatch most closely maps to what we now call a Jack Russell, and it makes perfect sense, really, because he was a ratter. Now, why people have been confused is Jack Russells often have legs. Hatch had normal length legs. But if you go back to older depictions of Jack Russells, that seems to be a fairly modern occurrence. Older Jack Russells had longer legs. But we also found out one other thing that wasn't expected from the SDR analysis. One of the things that SDR analysis can tell you is whether this individual is male or female. Now, everybody had assumed Hatch was female because there was no baculum. Many mammalian species, not humans, which is why we find this a little bit strange, but it's actually more common than us. The male has a penile bone, which is called a baculum. So gorillas and monkeys have it, it's humans who don't. That's why we're weird. There was no baculum found with Hatch's skeleton. So the assumption had always been Hatch was female. But our DNA test conclusively showed Hatch was in fact male, which means at the bottom of the solent, a little bit of Hatch's penis is still lying, I'm afraid. It was never recovered with the rest of the Merry Rose. So the secrets of Hatch. Hatch had light brown fur. He was a carrier for a genetic disorder that we now see in some modern breeds, including Jack Russells, interestingly enough. Unexpectedly, Hatch was male, not female, and he is most closely related. We can't say he is the same breed because that just didn't exist 600 years ago, but he's most closely related to Jack Russells. So we completed that work around 2014, and even as we completed it, it was an old-fashioned approach. Svante Pablo, in 2009, 2010, had developed a technique using next generation sequencing. So you know how I said Sanger dideoxy sequencing, that's how we read our little bits of code? There's an alternative technology called NGS that allows you to read the entire genome of an individual in one go. It's quite expensive, costs thousands, but the entire genome. We read little tiny bits of the genome because that's all we could do. But there was a technology to read the entire genome in a single go. And the genius, as Svante Pavlov did, was he was able to apply that technique to incredibly old samples, to things that was just simply beyond consideration that would be possible with Neanderthals. I mean, this is really interesting. So this is, I used to teach this. I used to teach that there was no obvious crossbreeding between human, Homo sapiens, us today, our ancestors, no obvious crossbreeding between our ancestors, and Neanderthals, which is a related cousin species which existed 40 to 50,000 years ago. Svante Pavlov clearly showed there was crossbreeding between Neanderthals and our ancestors. In pretty much a large majority of the human race, you will have somewhere between 3 and 6% DNA descended from Neanderthals or one of the other non-Homo sapien hominids. Now, when Homo sapiens, us, evolved, there were multiple hominid species a bit like us but different. Some of them, their DNA is still present in modern populations. It's in me, for instance. I know there's Neanderthal DNA in me. He was able to do something else even more amazing. He was able to identify what they call ghost populations. Populations there had been no archaeological or anthropological evidence for, but we know must have existed simply from ancient DNA analysis. Whole hominid species. And it was just so much better. So not only could you get this vast wealth of information, the whole genome in a single read, but it helps clear up so many problems that we had as well around the problems of contamination. I won't go into too much detail here, but basically the way the NGS works means you can look at multiple reads at once and you can very quickly see if you have a contamination issue. So not only did it give you so much more information, it also solved one of our big problems, the problem of contamination. Now, that was amazing. So we thought we'd have a go at that. And we launched in. We launched in feet first without probably doing all the necessary reading. So we thought we'd try and sequence one of the crew of the Mary Rose. So we took a bit of a tooth from a crew member of the Mary Rose and we crushed it up, we extracted the DNA and we used next generation sequencing technologies to study it. What we found is that only 1% of the data we got back was human. Only 1%. Now, this is actually a common problem with ancient DNA. It's a consequence of the degradation of other things being in there, such as bacteria. But there's so little of the DNA that actually comes from the original individual, you only get 1% of the data is usable. Now, that means if we just redid that experiment 100 times, we would probably be okay. But that would cost £100,000 to sequence one individual. That's just not possible. So the way around this is what they call capture technologies. You extract all of the DNA. Remember, only 1% of it is any good to us. Only 1% of it can be used. And we use capture technologies to pull that 1% that we need out and leave the other 99% behind. So that's called baiting. So we repeated our baiting, and it went quite well. We haven't published that data, but it has worked. This slide just shows how baiting works. It is actually quite technical, so we'll just skim over that. The idea is, though, we're going to use baits. We capture just that 1% you need. So this would be the modern approach. If I had to do hatch again, this is the way we would do it, right? There'd be none of that mitochondrial, there'd be none of that PCR, there'd be none of that Sanger sequencing. We would extract DNA. If I had my way, I wouldn't use a tooth. I'd smash up the skull and get the pectorus bone out. It'd give me a lot more DNA. I'd make what's called an Illumina library. I would then bait that library to get the 1% we need. I would then put it onto a machine called a HiSeq or a NextSeq, which would do the massively parallel next generation sequencing that sequences the whole genome in one go. Then I would give it to my colleague, Sam Robson, who's an expert in bioinformatics, and he would map that, identify all of the SNPs in the genome. He would validate it with a C to T transitions, which I won't talk about. And you would create a complete digital record of that individual's DNA. So that's how I would do it now, and that's how we started to do the crew members of the Mary Rose. But it is still early days. I'll just finish up with some acknowledgements and that. So that picture at the back is one of my dogs. That's called Lady Snufferton. She's a terrier cross with a lot of different bits of DNA in her, so you can tell from her face there's a lot of stuff going on there. And I would like to thank some people in particular. I worked very closely with Sam Robson, all the human crew. It would be impossible. The whole next generation sequencing thing would be absolutely impossible with Sam Robson. And in fact, because of his skills, he leads this project now. There was a technician called George Zougannalis, who has now moved to another university, but still works on ancient DNA. Niru Naha, who is my technician, I've still got, and she's still with me. Mazir and Bonab were at Portsmouth. Both have moved on to other universities now, but I keep in contact with them. So that was the UOP team. Gregor Larson and his postdoc, Ross Barnett. Gregor Larson is at Oxford at the moment, a place called the Paleo Barn, and he's probably one of the cleverest people I've ever met. He's slightly scarily clever. A guy called Arman Ardalan, who helped with the mitochondrial work, which actually didn't go anywhere, but when we started, we didn't know that. Rob Ogden, who's an expert in animal genetics and animal SDR. David Radford, who helped with doing that tooth. I really like that tooth. So thank you very much to David. He's a dentist at KCL. And obviously the Mary Rose Trust, Alex and Mark, who helped a great deal and supplied us with the material. And right back at the beginning, I said, well, I didn't start off being interested in ancient DNA. A colleague contacted me. His name was Glenn McConaughey. Very sadly, Glenn died in 2012 before the completion of this project, but it was Glenn McConaughey who first got me interested in ancient DNA. So that's the end of my talk. Thank you very much.
Thank you very much, Gary. That was really fascinating, and I must say you have a talent to explain very complex research in simple terms. And this is something I always say to my students, you know, when you meet someone like that, it shows that the research and the work behind that is really massive, is really great. Thank you so much as well for acknowledging a number of people. It's very much teamwork, and we know you are a very active member of the Culture and Heritage Hub of the University of Portsmouth. So thanks very much again for that. I'm going to use my privileged right as a chair of the session to ask you the first question, and I see that we are receiving a number of questions. My first question has to do with the very interdisciplinary, multidisciplinary character of your work. You alluded to the fact that obviously this technique was used by archaeologists and a number of other scientists. So how do you approach that, the multidisciplinarity?
It's always quite challenging because people have, I mean, when people are specialists, you get used to speaking to other specialists in your field. So it's always quite challenging. And I think you just have to be more open and not afraid to ask, well, slow down, I'm not sure what you mean by that. You know, this is not my field, what do you mean? But if you have a genuine interest in the topic, you want to learn, right? So, I mean, it's a truism, you know, we're always learning. And by being involved in multidisciplinary projects, I think that really is true. You are always learning because you're outside of your own speciality. So I guess if there's a thing I would say, and that's be keen to learn, you know.
Yeah, absolutely, and that's another of your talent and something we can carry with us. You know, be keen to learn, be curious, and to go and see people from other disciplines. So we have a question from colleague Joy. A really nice talk, Gary. Thank you. As you mentioned, DNA sequencing technology is rapidly evolving. Do you think the limiting factor is now finding high quality DNA samples?
Yeah, I do think that, and in fact, that touches upon a thing that's been going on within the archaeological community, where some of the big DNA sequencing labs, ancient DNA sequencing labs, have been hoovering up all of the good bones, basically. So we are very lucky to have access to the Mary Rose we work with closely. So that's an excellent resource of bones. And the DNA in them is very good quality because it's been in a very good environment at the bottom of the sea, very cold, protected from background cosmic radiation. So good quality DNA. And we've also got a relationship with the Fishbourne Roman Villa as well, where we're doing some work there. The DNA quality probably won't be as good, but the samples are very interesting. So there is a real issue of finding good samples, you know, which haven't already been swept up by the big labs.
That's really interesting. Maybe I follow up as someone who's really not from this field. Is there some sort of database or something like that? And what's the regulation in this matter? Can you source DNA from anywhere? How does it work?
So you would there are databases of the DNA once it's sequenced and analysed. That goes up into public domain databases usually. But to get the samples, you would have to normally approach the museum or the owner of the bones, and most of these samples would be considered relics of cultural significance. So there's an ethical approval process. So we did some work with the Natural History Museum on some old bones there. So that can be that took nearly 2 years to get ethical approval from the Natural History Museum. The Mary Rose relationship was much quicker. It only took like 2 or 3 months. So Fishbourne we're still trying to sign off on. So you approach the museum. There's some paperwork to be done. You know, the destructive testing has to be signed off on by the museum as ethically okay, and then you can start work. Once you've got the data, that data is uploaded to public domain databases.
That's great. Thank you very much, Gary. I go back to the ethic probably at one point, but we have a question from Karen. Fabulous, Gary. You made it seem so straightforward. It is difficult to think of a question. What would you do differently if starting again or taking on a new project, or what has changed that would make your approach different now? And she goes on asking, which crew member was it that you tested?
Yeah. So we're working on several crew members at the moment, and we have now actually developed more of a pipeline for the process so that we can do things quicker. I think I would still probably take the teeth as opposed to the petrous, because I'm a bit concerned about trying to get the petrous bone out without damaging the sample. But we can take that tooth and we can now scan that tooth, CT scan and image scan, so you've got a permanent digital record of the tooth. Because DNA testing is destructive testing. Once we've got the tooth, we have to destroy it. But we can take a digital record of it, and that would be one of the things I would do it that way, as opposed to having a physical record of the false tooth at the moment. Yeah. Then I would also, as I said, probably I would do an initial test to make sure that DNA is of a quality that's usable by using PCR and Sanger sequencing. But once I've confirmed that the DNA is of good enough quality, I would go straight to the NGS. Really, I think it's you just get so much more data, and it's so much more robust in terms of checking for contamination. So,
thank you very much.
The two individuals we did were the Mary Rose is interested in, because some of the individuals may not have been completely European. There were some suggestions they may have been from North Africa.
Right. So that's really fascinating, and this leads me to my sort of ethic question. I'm aware of the fact that in a number of countries, DNA tests are not allowed. There are different reasons for that, but I suppose one of the reasons is also because it's a Pandora box, isn't it? Like you're going to find crazy things or things that could be interpreted in a way or another in terms of sort of national unicity of the population, or patriotism and so on and so forth. How do you approach that actually?
Well, I've not really come across that. I mean, there are lots of different types of DNA tests you can do. That SDR test, for instance, is the one that's used by the police service for identifying criminals in this young country and America and across Europe. That test doesn't give you any phenotypic information. It's almost designed deliberately not to give phenotypic information, right? So you can't say from that SDR test that this person is 6.5 foot tall and of this ethnicity. Yeah. All it does is says that this person matches up to this sample. The blood found at the crime scene matches to this person. So that, I think, is less controversial. Even that, there's controversy about who should be on the database. Should you be on the database, I guess, if you've only been charged with a crime? Or should you be wiped from the database if you're acquitted of the crime? There's some areas of debate about that. In terms of where probably the biggest ethical dilemma is at the moment is if an insurance company says to you, I want you to have a health test to see if you're prone to a genetic disorder, and if you are prone to a genetic disorder, we're going to pump your premiums up. There's a lot of debate about that at the moment. So I guess at some point these discussions will have to be had in governments and between administrators.
Absolutely. Are you aware of private, like purely private institution doing similar research as you're doing?
Not on ancient DNA, but on modern DNA, yeah, you can find these. The two obvious ones are Ancestor.com and 23andMe. There's a lot of fly-by-night, really dodgy ones. There's loads of dodgy ones out there, but Ancestor.com and 23andMe, they give high-quality data. But that's on modern samples, yeah.
Yes, on modern sample, not on ancient, for the reason you explained before, and the fact that you have to go through a number of procedures and ethical procedures.
Yeah, it's much more difficult, yeah.
Yeah, that's great. So what's the next step for your research then? What are you going to study now?
Well, as I said, we're working. We've trouble is it's quite expensive, but we've started working with the Mary Rose again on some of the sailor crews, the crew members, particularly some of the bones have got all muddled up. So there was like a gunnery crew where you don't know which leg belongs to which arm bone. So they can resolve that so they can reassemble the skeletons. The other thing is the Fishbourne Roman villa, which we just started work on, and I'm quite keen to go back and visit some bones that I've got, which when I tried maybe 6 or 7 years ago, I didn't get to work. And these are particularly some bones from Canterbury Archaeology Trust. Didn't get to work, but I think I didn't get to work because I didn't know as much as I know now. So I think if I can revisit those bones, I think that there's a much higher chance of it going back. So maybe some continuation with the Mary Rose, maybe some new stuff with Fishbourne Roman Villa, and I would like to maybe revisit some things that failed in the past, but maybe we can get to work this time.
Excellent. I love the fact or the idea when you say that you want to revisit bones. I have no idea what you mean, but
we keep the bones in a special storeroom, and they're all catalogued now. My technician catalogues them with a code number, so we drill them, and then we try to extract the DNA. If we can't get it to work, we just keep the bone. We like the bone powder, so we can go back to the bone powder and try again. Yeah, I don't go back to the burial site.
It makes it look so scary. All right, Gary, thank you so much. It seems that we don't have much more questions. That was a fantastic talk as usual. Thank you for being so easy to understand and for bringing that much science in a very simple manner. That's fantastic. As you know, our webinars are always recorded, so you can always find them on the website of the University of Portsmouth under the banner Research Future. We have a new webinar next week with a colleague from India, Bangalore, who's working with me on question of labor and modern slavery. So tune in next week, Wednesday, 2 p.m. And I'd like to thank the team as well for supporting us, in particular, Gloria, her, Claudia, and Olga. So one more time, thank you very much, everyone, and I shall see you next week, 2 p.m.